Okay, this is Chapter 5, Part 2. So, synthesis and assembly. So, synthesis meaning making the parts, and assembly is putting those parts together. So, they divide that into two parts, the early phase and the late phase. So, synthesis of the viral parts is when all the parts of the virus are made. The viral DNA enters the nucleus where genes are transcribed into messenger RNA. RNA transcript moves into the cytoplasm to be translated into viral proteins needed to replicate the viral DNA. When the assembly happens, that's the late phase, the virus is assembled or put together. Parts of the viral genome are translated into proteins required to form the capsid and other structures of the virus. The new viral genomes and capsids are assembled, and the mature viruses are released by budding. The release, so the number of viruses released by an infected cell is variable, and it's controlled by the actual size of the virus, because only so many humongous viruses, as humongous as they can be, are going to fit in any particular cell. And then also the health of the host. And you can see some numbers here. I'm not going to go over those. But if even a small number of these virions happen to meet another susceptible cell and infect it, the potential for rapid viral proliferation is immense. Keep going here. You want to come back and watch this video. It's kind of got some cool animation in it. Damage to the host cell. So when this virus infects the cell, what happens to the host cell in the end? Well, CPEs, or cytopathic effects, are viral-induced damage to the cell that alters its microscopic appearance. Types of CPEs include gross changes in the shape and size, development of intracellular changes, inclusion bodies, and then sometimes, most of the time, accumulated damage from the virus. Infection kills most cells. Provirus is a persistent infection in which the viral DNA is incorporated into the DNA of the host. So I'm going to read that again. This is a persistent infection in which the viral DNA is incorporated into the DNA of the host. It may remain hidden in brain cells for many years, causing progressive damage and loss of function. Chronic latent state is when a virus goes into a period of inactivation in cells. You guys probably have seen lots and lots of commercials related to a very common type of virus that goes into the chronic latent stage, which is chickenpox and shingles. It can emerge periodically from various stresses in the body. One example is herpes simplex virus, which are cold sores and genital herpes. And then there's herpes zoster virus, which is chickenpox and shingles. And basically, what happens is, when you get chickenpox, if you've had chickenpox, that herpes zoster virus can live dormant in your nerves and later on rear its ugly head as shingles and cause you lots of pain. I remember hearing people talk about having chickenpox parties. So when their kid back in the day, and I say back in the probably 60s and 70s, I would guess, when their kid would come down with chickenpox, then, the parent would invite all the kids of the neighborhood over, and then they'd have a chickenpox party, and all the kids would have chickenpox. And, now we have, lots of issues with kids getting shingles. I'm sorry, people, it doesn't really matter what age you are, you can get shingles. And that's only if you've had chickenpox. So if you've had chickenpox, you are susceptible to shingles. And definitely emotional stress and physical stress can cause it to rear its ugly head. So viruses and cancer. Oncoviruses are viruses capable of initiating tumors. So about 20% of human cancers are caused by viruses. Transformation is the effect on the cell caused by oncogenic viruses. It can cause an increase in growth rate, alterations in the actual chromosomes, changes in the cell surface molecules, and the capacity to divide for an indefinite period, which is not a good thing. Cancer-causing viruses include the human papillomavirus, the Epstein-Barr virus, and the HTLV1, the human T-cell leukemia virus. So feel free to, read about these outside of this PowerPoint. You could learn a lot of interesting things in there on those. 3 mechanisms for viral induction of cancer. I'm not going to really get into this deep, so I'll let you read over that on your own. We're going to talk about this later on in the semester. So viruses that infect bacteria, specifically the bacteriophage, discovered in about 1915. Its phage means eating, so most contain double-stranded DNA when we're talking about bacteriophages. Every bacterial species is susceptible to specific bacteriophages. So remember, these are viruses that infect bacteria, okay? So they often make the bacteria they infect more pathogenic. So what are they, right? Well, a T-even bacteriophage, we're going to talk about this in depth from here for the next few slides. They infect E. coli. So I'm not going to get into the shapes, but if you look at it, if you look at this T-even bacteriophage, this is a virus that infects an E. coli cell. Okay, and it kind of looks like it's from another planet. Looks like it's something that might be floating around in space, right? So we have the head, we have the collar, we have the tail pins, okay, that actually are part of what stick into their victim, and then we have these tail fibers. Okay, so when we're talking about the the T-even bacteriophage, we have a couple of different life cycles that we want to talk about. So the lytic life cycle is actually, well, I'll get into this step by step. So they adsorb to the host bacteria using specific receptors on the bacterial surface. So of course there needs to be some sort of receptor there. The nucleic acid is then penetrated, or the nucleic acid penetrates after being injected through a rigid tube inserted through the cell wall and membrane. So I think of this this T-even bacteriophage as, when we go back to the picture, it kind of, comes in contact with the cell and then pops itself down into the cell membrane or cell wall and injects its DNA into that cell. Okay, so the nucleic acid penetrates after being injected through that tube. The nucleic acid entry causes cessation of the host cell DNA replication. So that entry of the T-even bacteriophage makes the host cell stop replicating its own DNA. Because it's injected into the cell, there is no uncoating step as there is in the animal cells. The viral replication and synthesis of viral proteins are completed using the host's machinery, and parts spontaneously assemble into the bacteriophages. Okay, so that's the lytic life cycle. Okay, a little bit more on that. An average size E. coli cell can contain up to 200 new bacteriophage units. Eventually, the cell becomes so packed with viruses, it will burst, releasing those bacteriophages. And upon release, those virulent phages can spread to other susceptible bacterial cells and begin a new cycle of infection. So, the lytic cycle has adsorption, penetration, duplication. There's more here on the next page. So it sticks to the cell, pokes through the cell, injects the DNA into the cell, and then there's a duplication of the phage components using that DNA code. Okay, and then there's the assembly of the new virions. They mature, and then the cell bursts. Okay, so that's the lytic cycle. Now, lysogeny, or the lysogenic life cycle, is the silent infection. Okay, on that same chart that I just kind of showed you in the pictures, there's kind of an offshoot step. Okay, so in the lysogenic life cycle, the bacteriophages undergo adsorption and penetration. So the actual DNA is injected into the cell, but they don't undergo immediate replication. The viral DNA enters an inactive prophage state and is inserted into the bacterial chromosome. So the DNA of the virus kind of is incorporated into that bacterial chromosome, and then each time that bacteria replicates or duplicates itself through binary fission, it is copied. That virus portion of the DNA is copied during that normal bacterial cell division. And so when we talk about lysogeny, it's a condition in which the host chromosome carries that viral DNA and replicates it with its own DNA as it multiplies. Okay, so induction, viruses in a lysogenic cell become activated and progresses directly into viral replication. And then lysogeny is considered the less deadly form of parasitism because it allows the virus to spread without killing the host. So scientists believe there are more bacteriophages than all other life forms in the biosphere combined. So you can take a look at that video in the regular PowerPoint. So I just want to go back to this. So this is the lytic cycle, and then this illustrates the lysogenic state, okay, the lysogenic cycle. So we have adsorption, we have penetration, but then instead of a duplication of the components, that viral DNA right there is incorporated into the bacterial DNA. And then eventually, when the time is right, it will start to replicate the genetic material and then move back into the lytic cycle. So it can actually go from, the beginnings of the lytic cycle to the lysogenic state and then back to the lytic cycle. Okay, so watch those videos. Those will help you understand that a little bit better, as well as reading your textbook. All right, so characteristics of prions composed entirely of protein. These guys don't contain a nucleic acid, and there's lots of questions about prions. Definitely reference your book on these. There's a little bit in the book on these. Viroids, these are virus-like agents that parasitize plants. Okay, and of course, it's significant in, with farmers and such that are taking care of our food. Viruses and human health, common causes of acute infections, colds, hepatitis, chickenpox, influenza, herpes, warts, prominent viral infections worldwide, dengue fever, Rift Valley fever, yellow fever, and so on. So I'm not going to read through all this, but, the book has some more information on this. Treatment of animal infections. So antibiotics designed to disrupt prokaryotic cells have no effect on viruses. So when you go to the doctor and, you have a sore throat and you feel like, the Sussex County funk is going to kill you, then, they're not necessarily going to give you antibiotics. Obviously, if you start out with a viral infection, it can kind of end up turning into a bacterial infection that is treatable, but it's usually having to do with stuff kind of hanging out in your sinuses or something like that. So antiviral drugs that block viral replication target the function of the host cells, and sometimes these can have severe side effects. Interferon, which naturally occurs in human cells, is used with some success in treating and preventing viral infections. And of course, vaccines are the best way to treat or prevent, rather, viral infections or viral diseases. So there's a video there. You want to take a look at that video. And that's it. So that is the end of Chapter 5. Don't forget to do your homework on Connect.